I'm Bruce Bugbee, and I am pleased to be presenting this seminar to you in Horticulture Department at the Texas A&M. I'm pre-recording this here in our studio in my laboratory at Utah State University because I hope I can show you how compelling seminars can be when you can interact with the screen as opposed to just showing slides. So we'll record this a few days early and then I'll take questions after the end of this. So I've chosen this as a topic, this turning photons into food. And this is a title I've used in other seminars. First of all, I love the alliteration of the title, but it also leads to measurements and models. And in this seminar, I want to talk specifically about how those work in horticulture. I need to acknowledge two major funding sources for this before we get started. One is NASA. I have been very fortunate to be funded by NASA almost continuously through my whole career. And it's the ultimate in controlled environment agriculture. We're working on closed systems for Mars and regenerative systems for growing food. And this model that I'm going to talk about is really applicable to that situation. In addition to NASA, we're also funded by the USDA. And this is called the LAMP project, Lighting Approaches to Maximize Profits. That means supplemental lighting in indoor agriculture and in controlled environments. So some years ago, I was fortunate enough to get elected as a fellow in the American Society of Agronomy. And I've regularly attended their meetings for many years, and I hear talk after talk of models for agriculture, for field agriculture. And I want to compare and contrast those models that agronomists used to the model I'm going to talk about for more controlled environments and for higher value crops. So that's a little bit of a prelude. But first, so if I'm giving you a seminar, one of the things that's a great joy of my life is going to college campuses around the nation and getting to experience the campus. And you see the students, you get the atmosphere. Campuses are all very edifying places. And I have been on the Texas A&M campus twice previously, both times to give seminars. And they were both 12, 15 years apart. One of them was in the agronomy department. But as part of that, of course, I looked up your department on the internet, and you don't have to go very far to know the rich heritage of Texas A&M, because it's one of the oldest land-grant schools. So here's the picture of the campus, 1883. That's the whole campus right there, the mess hall, the mechanical hall, and of course then I thought, well, that's a nice picture. The field's right next to campus, and then I looked for an updated picture, and it's pretty much the same except for one big building right in the middle now. So it tells you the importance of horticulture on the Texas A&M campus. You are one of the few pure horticulture departments left. Most schools have merged horticulture with agronomy and a lot of other departments. So there's a proud heritage. I've worked with Fred Davies here, and before Fred, Malcolm Drew, some really key people in your department. It's been an exciting collaboration. And of course, on our faculty, David Hull got his PhD in horticulture at Texas A&M. So let me start with what I like to call the 9 cardinal parameters of plant growth. Go one more slide back. This is, I've seen other people use this concept in this slide. You know, this slide is memorable and it sticks. But we have 5 parameters in the shoot, 4 in the root zone. When we, in any one of all of these, we modify to optimize plant growth. When we go to those models in agronomy, and let's see what color we got here. Pen, let's go with light blue. A huge number of those models are on water. Water is a limiting factor in field agronomy, and year after year there's symposia after symposia on the efficient use of water. This is the most limiting parameter. Out by Texas Tech, boy, they're all trying to grow cotton with less and less water. It's a huge thing. In addition to water, there's numerous symposia on nitrogen. That's another key parameter in field agriculture, nitrogen and phosphorus. But in high value horticultural crops, everything at the bottom is a minor input compared to light. And now I should put that in yellow because we're talking about light. Light drives everything. For field agronomists, the sun comes up in the morning and you get what you get. You can't change it. Their models are all for summer conditions, where there's lots of light in the summer. Our models look at horticulture models growing crops year-round, when the light is not at all optimum in the winter, and it's very limiting to this whole system. But if we're growing a very high-value crop, we can afford to modify all these things. We give them plenty of water, plenty of nutrients, fluff up the root zone so they get plenty of oxygen. How about root zone temperature? I got to get a different color for that. How about, we'll go with blue. Guess what? We put on mulch for high-value horticulture crops, so we end up modifying root zone temperature too. So it's all based on trying to optimize light. When we analyze this, back to yellow, in this model, we're looking at the efficient use of every parameter. That's what models help us with. What are the inputs? What are the outputs? So efficiency of a system is the output over the input. Very simple. And to make this work, you want the same units in the input and output. But in this case, the efficiency would be when they're the same units, but the efficacy of a system is when they're not. And we can quantify inputs and outputs. So in this model, I will attempt to derive from first principles what the maximum efficacy of a horticultural system can be. And the output we're going to do in grams, and the input we're going to do in moles of photons. Light in, biomass out, and when all the dust settles, 1 gram per mole. Remember that number. It's as simple as it can get. How do we get that number after all these things derive together? I'm a big fan of simple models, the ones you can do on the back of a napkin in a restaurant. And that's one of the joys of my life, is sitting around doing math on napkins in restaurants, because they're simple models, so you can do them. So let's take a look at this. Where does this come from? First of all, I want to give tribute to a key player in my life, and it was Robert Lumis, who was at UC Davis. I had the great honor of working with him. He died just 5 years ago. This is one of his key papers. And look, 1963, it's an old paper, maximum crop productivity. He was in the agronomy department. He did model horticulture crops, but he was focused more on agronomic crops. This is Crop Science issue 3, so it's one of the early versions of that. To assess factors that limit productivity, we need an estimate of potential productivity. That was his goal, not what does nitrogen and water limit it. Estimates of yield potential have been done before. We calculate yield on a quantum basis. And by quantum, he means moles of photons. Moles of photons in, yield out. And our choice, now look at this, our choice of 400 to 700, now look at this unit right here, MU. This was 1963. SI units weren't widespread yet. This means millimicrons. So 10 to the third times 10 to the minus sixth, that is now a nanometer. We would never get a paper published calling it a millimicron. 400 to 700 nanometers. That's traditional photosynthetic radiation. We're going to come back to this in this seminar based on the classic work of Emerson and Lewis with Corella and algae. So he's counting 400 to 700 nanometer photons and looking at photons. I got to tell you a story about this. So I'm a young graduate student just barely learning things, and you'd go into his office. He had a big whiteboard, and there were personal computers, but they were really rudimentary. But you would kind of humbly ask him some question, and he'd say, we can calculate that. And he'd jump up and he'd go to his whiteboard and he'd write all this stuff out, and he'd say, let me see, we can assume this, right? And then he'd be talking to you like you were a colleague, and you're watching it and you're too intimidated to take notes. You just try to watch the whiteboard, and then you go right back to your office and see if you can reproduce all the calculations you just did on his board. And that happened over and over again. I just loved it. He was a big influence in my life. And he got me onto these models. Now one more slide. 1967, he got into doing photosynthesis and plant communities, and look what he says in his paper. An immense number of calculations are required in such a model, but an IBM 7044 computer can do it in 6 seconds. So now it would be 6 milliseconds in our computers, but can I get rid of this? I'm going to put snooze. Okay. So, but back then, model computers were just coming on, and everybody had fun with their computers. So, we're going to start with first principles in this analysis. And the first principle is what I consider to be the most important equation in the universe, in the world. Equations are inputs, and then they go to outputs. So what do you think this equation is? If you show this to physicists, they say, well, it's E equals mc squared. How about to a biologist? No subscripts, very simple. We teach this in high school over and over again. Texas A&M probably teaches this in 30 or 40 different classes on campus and various analyses. By now you recognize this. This helps to know that this is carbohydrate, CH2O, and by now you recognize it as photosynthesis. It's the most important equation in the world. Think about this. This takes a low energy gas, we use it to put out fires, and water, the simplest of inputs, and it makes a very high energy solid and rocket fuel. The outputs are super high energy and the inputs are super low. It's amazing. That's why it's the most important equation in the world. Now, we teach this and we make students memorize the stoichiometry and various things. If you take a more advanced class, you say, whoa, we're missing something. You can't go from low energy inputs to high energy outputs without something pushing it across, and that push is photons. Now, carbohydrate is food, and now we're turning photons into food, and we're doing this quantitatively. This is 1 mole of carbohydrate, and this is 10 photons. Not just conceptual, but there's our first step in the model. 10 photons in, 1 carbohydrate molecule, 1 carbon and carbohydrate out. This has to balance, because remember, the first law of thermodynamics is energy in equals energy out. So with this, this has got to have plus heat over here. I'm running out of room. H-E-A-T, heat. In the outputs of this, then the whole thing balances. So now we can ask, what's the efficiency of this equation? How much is heat? Well, the heat's lost. We don't count that. And interestingly enough, when we analyze this, we don't count the oxygen. We just, oh yeah, there's oxygen. Well, wait a minute, you started with carbon dioxide. If we count oxygen, we get even more outputs to this. But all we're doing is the food out and the photons in. When you think about this, these low energy inputs, high energy outputs, we're misleading students when we write this equation like this. And we always do, write it like this. We should write this equation like this. We got real low energy inputs down here, and then and this should be in a 2-point font, and this is in 30-point font out here. Boom, all the way up to high energy outputs. So I think we should start changing all the high school biology books and start writing this equation like this. And then, to help the students understand energy balance, kaboom, we put 10 photons in here to push the whole thing up the hill. When we do this analysis for the potential efficiency, what do you think we get? The energy in the photons versus this? 30% is pretty close to the maximum theoretical efficiency of this process. And you think, whoa, this is the most important equation on Earth, and it's only 30% efficient? And this bothered me for a while, because this is my profession, and people that, you can imagine the state legislature, we're giving you guys a lot of money. How come you can't get this better than 30%? And I realized this is not one step for this thing to go to that. It's a lot of little steps. And that's why I show it as a dashed line. There's a lot of biochemistry in here. Step 1, step 2. The light reactions, the dark reactions, we're skipping a lot of things. So now when I talk about this, I say, this is 23 steps, and each one is 95% efficient, so this whole thing is 0.95 to the 23rd power for 23 steps, and that's 30% efficient. So now I don't feel so bad about photosynthesis for being 30% efficient. Let me work through the math in this. This is some real simple math, and once you see it, you'll get it. Remember, we have output. Let's see, where can I write on here? Output, I'm going to put output over input. The output is 1 CH2O, and the input is 10 photons. Now let's see if we can calculate the efficiency of this. 1 gram of sucrose, 1 mole of carbon in sucrose is 490 kilojoules per mole. You can look that up in a book. This is just sucrose now, the first step out of photosynthesis. 10 photons, if we do Planck's equation, which is E equals hc over lambda, and you can look that up, and you put 550 right here, which is the average wavelength of light from the sun, and you solve this equation, we get 217 kilojoules per mole of photons. Now these are just physical constants that we look up, but look at the math. Put it up here. 490 over 2170. I like this model because you can do the math in your head. You don't even need a napkin to do this. And now, well, that's about 500 over 2,000. It turns out this is close to 22 12% efficient. How come we said 30% here? Well, we're very clever as biologists, and we know that you can do photosynthesis with just red light. And red light, right here, putting this in another color, or try red, is if we put in 680 nanometers here, these photons have lower energy. And it doesn't matter, one photon excites one electron. What doesn't matter what color it is. And when we put 680 there, this number becomes 176 kilojoules per mole. Low energy red photons, and this becomes 1760, and this then it goes to 28% efficiency. Now this says 30, that's 28, but it doesn't quite take 10. That's more about 9 photons to drive this. But there's the math of calculating photosynthetic efficiency. First step of the model. Now this is the energy efficiency, and we want to know biomass. So let's take this another step. Quantum yield is moles of carbon fixed per mole of photons absorbed. And moles of carbon fixed, remember, is C, let's go back to yellow, is CH2O. That's 1 mole of carbon fixed, and here's mole of photons absorbed. So there's quantum yield. Now, this is a graph made by Shuyang Zhen, and she did that when she was a postdoc with us here in Utah, and now we're thrilled that she's a new assistant professor in the Department of Horticulture. So I'm going to show a little bit of her data here. Here's quantum yield, and this is yield, so it puts the carbon on top. The inverse of that is quantum requirement, so 1 over 0.1 is 10. So there's our 10 photons to fix CH2O. And that's the top line. And you can see nothing quite gets to 10, but it gets close. But you can also see that, here's the light. Photosynthetic photon flux density. As this goes up, this goes down. You can the system's only efficient when it's running slowly with low light. As soon as you turn up the light, the efficiency gets less. This starts to go down. The crop doesn't much matter. These are all clustered close together up here, and they have similar slopes. Grand Rapids lettuce is a very light green lettuce. You can almost see through the leaf, and that's a little bit lower, but this is also photons absorbed. So if it goes right through the leaf, we don't count it. We only count the photons absorbed. What about the purple one? Purple, I should have a slide of Marshall. That is full of anthocyanins. It's dark purple. It's prized for salads, but all those anthocyanins come at a cost. They're not photosynthetic, and purple Marshall takes a hit until we get high light, and now it starts to get close. So this is the photoprotective pigments become useful in very high light. So let's take this data now and start building our model. And what we're going to do is assume 400 micromoles. Now you can grow great crops with this. This is similar to the average through a day. And when we do that, we get about 12. So in a reasonable crop, we're going to model this assuming a quantum requirement of 12. This data was done on single leaves. It was done with the clamp-on photosynthesis system. If I say we did it, that means Shuyang did it. But it was done with a single leaf. But this is a fundamental principle. It's also true of whole plant canopies. We can scale in size from this data. 12 photons. Now let's go back to our model. CO2 in, carbohydrate. Now we're going to turn this to a yield model. Carbohydrate is 12 grams for the carbon, 2 times 1 for the hydrogen, 16, 30 grams per mole. So now we take 12 photons in and we get 1 CH2O out, but that's 1 CH2O is 30 grams. 30 grams out, 12 in, 2.5 grams per mole. Wait a minute. We just said the maximum you can get is 1. What's going on? This equation is only for photosynthesis. What happens at night? Respiration. This is only to make sucrose in plants. At night, that sucrose gets turned into proteins, lipids, cellulose for cell walls, all the other thing a plant needs to grow. And when we run this backwards, it's respiration. Now, I would argue that's the second most important equation in the world, running it backwards. And some of my microbiologists argue with me, and they say, no, respiration is number 1, and your equation that way is number 2. But all right, we'll debate that. But so we run this backwards, and whole plant growth gets less efficient. Here is a picture of some of our gas exchange chambers. This is, I think I can erase that. Look, eraser. I can make that part go away. This is some of our chambers out in a greenhouse. You can see all the tubing on here. We're watching the plants breathe. We're measuring CO2 in the day, CO2 out at night, back and forth. And with that, we get the whole carbon balance. The next step in this is called carbon use efficiency. And what this is net photosynthesis divided by gross. Now this part's gross photosynthesis here, with 2.5 grams per mole is gross photosynthesis. But when we take net, which is the weight of the plant after 24 hours, fixed it in the day, it takes a couple steps forward in the day, respires at night. Couple steps forward in the day, respires at night. We get 60% efficient. This is the carbon use efficiency. It's not exactly respiration efficiency, it's carbon use efficiency. So now, here's the day multiplied by the efficiency of night, 0.6 times 2.5, 1.5. Again, pretty simple math. That times that is 1.5. We're still not down to 1 gram per mole, which is what we said is about the peak you could get. This approach so far does not include step 1, I'm going to put 1 right here, which is photon capture. Photon capture. If a photon hits the ground, what's the efficiency of that? 0. It has to be absorbed by a leaf. So this is not 100%. A black hole would be 100%, but this is not 100%. Step 2 is this quantum yield. Step 3 is this carbon use efficiency. Here. The next slide takes these 3 components and shows what happens when we multiply them together. Here, before I do that, here's our new system. We're just building this. Shu Young used an early model of this, and she did such a great job. We all got inspired to make the system even better. So here's 10 chambers in here, and here's Noah Langenfeld with these plants. In this system, all this is a box, and you blow air in, and air comes out, and the difference is the breathing of the plants. It's measuring metabolic rate of the plants. If we take this on 24 hours, here's midnight to midnight, and here's daytime, PN, daytime, and here's respiration at night. This minus that is daily carbon gain. And then to take it a step farther, respiration, we assume, goes in the same rate, light in the dark. So gross photosynthesis is this whole thing. This whole area under here is all gross photosynthesis, and this minus this is net photosynthesis. That's how we calculate carbon use efficiency. And unlike some of the other parameters, boy, 60, we've had Jonathan France did his PhD in this lab on that exact topic, and we get 60% efficiency, and it's hard to change that. It's quite remarkable. That's a topic for another seminar. So here's the system, which we're getting ramped up to use. Here's the model. We call this the energy cascade model, current and potentially achievable yield. 1 mole of photons right here goes in, and we say current efficiency, meaning high end but achievable now. Fraction absorbed photons, 80%. That is not easy to achieve. When the plants are little, the efficiency is almost 0. Finally, it gets up to 95%. But this is the average over the life cycle, 80%. And that is a very good crop. Most of our egg systems can't get that high. Quantum yield, 0.06. Now, this is a little bit higher light, but a more typical. Respiration, 0.6. This is the part we were talking about just now. These two numbers here, photosynthesis and respiration, multiplied together. Now we have a third number, 0.8 for radiation capture. And then finally, the fourth and final component is called harvest index. And that's the ratio, I think most of you know, the ratio of edible to total. The roots usually aren't edible in most crops. The stem's not edible. And in a good crop, we can get up to 50%. This times that, times that, times that, successive multiplication, 0.014. Here's our 30 grams per mole equals 0.4. Now we were shooting for 1. But what can we do with these numbers to make this a bit better? This, by the way, is one of the early papers in my career. We published it in the journal Plant Physiology, exploring the limits of crop productivity. When I worked in this model, and we grew crops and measured these 4 parameters. That's now one of my more highly cited papers from a long time ago. Now let's look at what we call potential. If we could inch this up to 90, and we could get this up to 0.8, a little higher, it's mostly going to take lower light, and inch this up to 0.65, and a crop like lettuce, you can eat 80% of the crop. There we go, 1 gram per mole. So that's a very efficient system, but a potentially achievable system. And there's 2 values of this. One, if you measure the inputs in moles of photons and you measure the output, you get this ratio. But if you have a gas exchange system, you can measure all of these parameters independently every day of the life cycle. That 1 gram per mole comes from grams, I better put this over here, grams per meter squared per day of the output, the growth rate of the plants, and moles per meter squared per day of the input. And you can see how the units cancel, so you get grams per mole. This is what we call the daily light integral, moles per meter squared per day. So this is area of the plants, like pounds per acre, but grams per meter squared per day. It's a pretty simple ratio. And how efficiently are the inputs used to this? So take a look at this as it applies to indoor agriculture. Now, after we've said we could get to 1 gram per mole, if everything's just right, we got to have variable spacing, everything's right, 80% harvest index, but we can get there with lettuce. What if all crops could get 1 gram per mole? A potentially achievable. What does that mean for indoor agriculture? Now we're paying the electric bill for the photons and we're counting the food out, but it's a very optimal system, so we potentially could get to 1. This analysis, which we published in Nature, that's 2 years ago now, LEDs for photons, physiology, and food, looked at the economics of indoor ag. First of all, we assumed 10 cents a kilowatt hour, pretty reasonable assumption. Microgreens are wonderful. This is the cost of photons as a percent of market price. 1%. Microgreens are very cost effective. The electric bill is small. The microgreens are valuable. Now we go to lettuce and it goes up by a factor of 5, but still small. Lettuce is harder to grow, harder to capture all the light. Lettuce does not command the premium price that microgreens command, so the electric cost is bigger. Now this, by the way, is also using our most efficient LED lights. It's super efficient LED lights. This is all good. Now let's see, okay, what else can we grow? Boom, tomatoes goes up by another, almost a factor of 4. What's going on? Tomatoes aren't as valuable per gram dry weight as lettuce, and guess what? The harvest index of tomatoes is much lower. You got a lot of stems and leaves and you can't sell those to people, so it's expensive. But tomatoes, we've refined the efficiency, and even if we pay 18% for the electric bill, we can grow tomatoes. How about everything else? Let's grow broccoli, let's grow strawberries, general vegetables. The cost of the electricity is equal to the value of the product. 100% means $10 of electricity, you grow $10 of food. They're not economic. And this is even with the maximum potential efficacy that we could get from this system. So when people say we can feed the world with indoor agriculture, they should read this paper in Nature, which is now, I'm pleased to see, getting traction. Lettuce is a great crop. Maybe tomatoes sometimes, but I don't see it going to general vegetables. And now, the 2 staple crops in the world are rice and wheat. Those 2 crops provide something like 80% of the world calories. Let's try to grow those under electric lights. 100 times. The electric bill is 100 times bigger than the value of the rice and wheat. It's just enormous. The reason this goes up so fast is twofold. Market price. It goes from high to very low. Now this is for dry weight, right? This is, we can sell the water in microgreens and lettuce and charge for it, so they're super high value. Down here they're sold dry. And on top of that, harvest index. These 95, 90, and then we go down much below 50. So those 2 parameters mean that it's very difficult to grow more complex crops under electric lights. In spite of what people tell you and all the money going into venture capital, it's not going to happen. These are the basic rules of physics. They're involatile. This is a review of some work that Shuyang Zhen did here in the lab, and it's on far-red photons. Now, if you've listened to Shuyang give a seminar, you already know about this stuff. To me, this is fundamentally groundbreaking research on photosynthesis. Here's a prism. The far-red photons are right here. You just barely can see them. And here's Shuyang, now in your department. I just have a couple slides on this. 1 photon excites 1 electron, and most people show a lot of biochemistry, and books show that. I'm more of a physics person, so I like a little physics diagram. This is the Stark-Einstein law. 1 photon excites 1 electron. So here they come. Boom! 1 photon hits this guy. He hits this little catapult, and he excites 1 electron. This is actually 4 photons, but he excites 4 electrons up to this worker, puts it on a mill, and it goes downhill, and it makes ATP. Boom, another photon hits this guy. He hits his launcher and up here, puts it in here, and we have NADPH. Now, there's a whole semester of biochemistry explained in one slide, real simple. Water gets split right here to oxygen. Here's the thing. This guy responds to far-red photons, and we have known this, but if you put far-red photons over here, the system doesn't work because this guy does nothing. You got to have photons, visible photons here to get the whole system working. So these are synergistic photons with these. And for decades, we've said, back from Lumis and Williams, 1963, we counted 400 to 700 nanometer photons. That's the yellow arrows here, 400 to 700. But what happens if we give them some far red? And now this is a very big deal because there's a very, very efficient LED that only puts out far red photons, 730 nanometers. If that works, those are low energy photons, which is why that LED is so efficient. Can we give them more far red? This has been known for half a century as the Emerson enhancement effect. And as Shu Young will tell you, Emerson tragically died in a small plane crash and never finished his work. His students didn't finish up, but he was looking at the value of these far reds in the 50s. One slide summarizing. Shu Young has two papers from here, one in Plant Cell and Environment and one in Frontiers in Plant Science. These are long titles, but you can pause and read the titles or go ask her for reprints. Here's the data from this gas exchange system. The plants are breathing day and night, in and out, in and out. Here's a blow up of it. Here's the plants. One of these had far red photons. One had no far red. See, those plants are bigger? That's the orange line. They only got 300 micromoles of visible photons, and the control, white light, had 350 of visible. So we grew, Shu Young, grew a bigger plant with technically less PPFD. But this one, she gave it 50 micromoles of far red. So they were equal if you counted 400 to 750 nanometers. The two treatments were equal. She measured photon capture in this. These are whole canopies of plants in that gas exchanger chamber we looked at. Here's the curves. This is pixel imaging from overhead pictures, which is tremendously powerful. We do that. We should do it more. Little plants, they get bigger. The pixels got bigger as they grew. The far red plants got bigger faster. And this is measurements of canopy photon capture with light bars above and below the canopy. See how they got to 0.8 near harvest? And that was what we said we could get to for an average before, but you could only get this with variable spacing to space the plants apart. Here's photon capture. Here's quantum yield. Remember 0.6? There it is, 0.6. Both of them were virtually identical for quantum yield. Far red photons were just as good as visible photons. For lettuce, and Shu Young's done this now on around 20 different crops, and far red consistently is, photons are equal to visible photons. Carbon use efficiency, here's our typical 0.6. These lettuce plants on the early stages started a bit small and climbed up. And some of this is seed respiration, but this is that ratio of day to night. So there's 1, photon capture, 2, quantum yield, 3, carbon use efficiency measured in a gas exchange system. And it shows the power of this model, both the theoretical parts of the model and the practical parts of it. I'm going to stop here on this slide. I'm happy to take questions. I'm glad I got to summarize some of Shu Young's data. I know she's going to keep working on this. Thank you, and I'll take questions.